Hello, my name is Emily, and these are my teammates Kelly and Jerry. Our project is a simultaneous detection of genetic and epigenetic DNA modifications by targeted next-generation sequencing for cancer screening. Cancer is the second leading cause of death in the US, with over 500,000 deaths projected overall in 2015. And the success of cancer treatment depends on the early detection of cancer. In fact, over 50% of cancer deaths can be prevented if cancer is detected in the early curable stages. It is also known that cancer is a disease of the genome, resulting from various genetic and epigenetic DNA modifications. If we can detect these modifications that contribute to the onset of cancer, we can detect cancer early. Also, cancer is highly heterogeneous, meaning that a single marker does not provide sufficient sensitivity for early detection. So there's an urgent need for a sensitive cancer screening approach. And our strategy is to develop a sensitive technology platform to simultaneously detect both genetic and epigenetic DNA modifications to improve cancer screening. Specifically, we focused on hepatocellular carcinoma, or HCC liver cancer. As shown in this picture, HCC is one of the fastest rising cancer incidence rates in the United States. The 5-year survival rate is 29% for local tumors and less than 3% for distant metastasis cases. Unfortunately, the current marker, serum alpha-fetoprotein, is only 40 to 60% sensitive. So in order to create our panel for HCC screening, we chose the most recent and frequently published HCC markers in literature. These include the genetic genes TP53, CTNNB1, and TERT, and the epigenetic genes GSTP1 and RASF1. However, using these markers is only practical if we can detect them in the periphery without a tissue biopsy. And our laboratory has shown that human urine contains low molecular weight DNA from circulation.
However, detection of this circulation-derived tumor DNA for sensitive cancer screening presents us with several obstacles. Because cancer is a heterogeneous disease, it requires a panel of multiple genes. In addition, for this method to be clinically applicable, it also must be cost-effective. And lastly, the DNA is short-fragmented and the modifications are only present in low frequencies in urine. So this requires a platform that's highly accurate and highly sensitive. Next-generation sequencing, or NGS, is a very powerful tool that's capable of multiplexing both genes and samples and capable of detecting specific sequences. However, NGS does not resolve all of our obstacles, and it has two major limitations. First, it has an inherent error rate of 1 to 3%. This makes it not sensitive enough to detect low-frequency modifications in the periphery. Second is the bottleneck of NGS data analysis, created by the millions of sequencing reads we receive from NGS. This is not only time-consuming, but requires high-level training in bioinformatics to analyze. So to overcome these obstacles, our approach was to develop a platform to increase the sensitivity of NGS for detecting low-frequency DNA markers in urine, and to develop a software to overcome the NGS data analysis bottleneck. We hypothesized that the sensitivity of a targeted amplicon NGS platform can be increased by preferentially amplifying the DNA modifications. For methylation markers, this can be achieved by designing primers to include the CpG sites, and for mutation markers, this can be done by incorporating PCR clamps to inhibit wild-type amplification. We also hypothesized that a customizable software that can accurately identify mutation and methylation for targeted NGS can be developed.
The conventional methods in detecting methylated DNA utilizes bisulfate-specific primers outside of the CpG sites. By doing so, there is equal amplification of both the methylated and the unmethylated DNA, so the sensitivity is not enough. Furthermore, this method is only applicable to greater than 100 base pair amplicons. In order to detect these markers in urine, less than 50 nucleotides of DNA needs to be detectable. So our method is to utilize a mixed YG primer design to move the primers within and to include the CpG sites. The YG primer design that we used includes that Y means C or T. And since C and G have 3 hydrogen bonds, while A and T only have 2 hydrogen bonds, when these oligos are mixed together, the methylation primer will have a higher affinity. So when methylation is present, it will be preferentially amplified. Therefore, when these samples are sent for NGS, most of them will be above the 1 to 3% NGS inherent error rate. To test if this is a viable method, we utilize the methylated septin-9 gene as our pilot study. Shown to the left, here is our NGS procedural flow. We start off with a methylation-specific PCR, as was described in the previous step, followed by an adapter PCR to add the adapter overhang sequences to the previous sequence. And lastly, we end with an index PCR to add barcodes, and these serve to differentiate among the samples and genes. To verify each step of the amplifications of the 3 steps, we conducted gel electrophoresis and confirmed the expected base pair sizes. So with that method, we tested a total of 150 urine samples, and we sent them off to NGS. Shown to the left is a manual data analysis method using a DNA visualization program called Genome Browse. This method took over 8 hours to manually analyze the data. And to the right, we see a comparison between our NGS method and conventional real-time methods. The yellow highlighted boxes show where methylation was detected by our method, but not by conventional methods, thus showing that NGS has a higher sensitivity than conventional methods. So with the success of our SEPTA9 pilot study, we can apply this method to our prominent HCC methylation markers, GSTP1 and RASF1. Shown here is an example of RASF1 and the amplifications from target-specific to adapter and to index PCRs.
So we accomplished our goal of detecting the methylation markers. However, we still needed to overcome the obstacle of detecting the mutation markers, TP53, CTNNB1, and TERT to 0.1% sensitivity. So our strategy to do this was by designing PCR clamps to inhibit the wild-type amplification and allow for the detection of mutated sequences. Previously, our lab has used locked nucleic acid, or LNA clamps, to inhibit the amplification of wild-type DNA and express the mutations for TP53. So we applied this method to CTNNB1 and TERT. However, the LNA was not potent enough. So we tested bridged nucleic acid, or BNA, to see if it would produce the same results. Shown in the gel picture to the right, we optimized the BNA conditions to successfully inhibit the amplification of the wild-type DNA. In the diagram below, the red regions indicate the BNA sequences, and the underlined show the expected mutations. So following the optimization of the target-specific PCR, we performed a similar process that we did with the epigenetic markers to the genetic ones to add the adapters and the indices. Looking at our overall NGS assay as a whole, we performed target-specific PCR to the genetic and the epigenetic, then we performed the adapters, PCR, and the index PCR, and we pulled all the samples together for a single NGS run.
So now we accomplished our first objective. In the meantime, we're developing a software that could overcome the NGS data analysis bottleneck. So the data received from NGS is composed of more than 20 million reads. Emily, in her SEPTA9 pilot study, analyzed this using Genome Browse in a process that took over 8 hours. So to facilitate the analysis process, we developed a program, Fast NGS Detect, that could automate the data analysis process. So the workflow of Fast NGS Detect starts on the left, with the files coming in as FASTQ files from NGS and uploaded to a cloud and aligned using Burrow's Weirdo Aligner, or BWA. The output from BWA comes in as sequence alignment mapping files, or SAM files. This file contains all the necessary information for NGS analysis, such as the reference sequence, the quality score, and the actual sequence of the read. Then the program enters the variant checking stage shown on the right. It checks the SAM file line by line and checks if each read is aligned to a genetic or epigenetic gene. If the read is aligned to a genetic gene, it calculates percent mutation. And if the gene is aligned to an epigenetic gene, it counts the number of CPG sites. For both genetic and epigenetic genes, the percent background mutation is calculated, which is the percent mutation inherently generated by NGS. All data is then organized and compiled into an Excel spreadsheet file. So to make sure our program was accurate, we performed software validation with our epigenetic SEPTA9 pilot study and our genetic TP53 constituted standard. So shown on the left, this graph shows the concordance between our Fast NGS Detect software and Genome Browse with R² close to 1. And shown on the right is the table of concordance between our Fast NGS Detect and conventional software. The results were consistent across both platforms. Overall, the time spent analyzing the SEPTA9 pilot study using Genome Browse took over 8 hours. However, using my program's Fast NGS Detect, the process was only 5 minutes. This is an improvement of 100-fold. Shown here is Fast NGS Detect's GraphQL user interface. The simple process consists of selecting the input files, output gene, output directory, and allowing the program to run. This program can be customized to include other genes and even other cancers. So now we accomplished both our objectives. So using the methods we described earlier for the genetic and epigenetic genes, we prepared 46 urine DNA samples and 5 reconstituted standards and sent them out for sequencing. The data from the NGS sequencing was then ran through Fast NGS Detect, and the data showed that we achieved a 0.1% sensitivity.
So in summary, we developed an NGS platform to increase the sensitivity of DNA modification detection to 0.1%. For epigenetic markers, this was done by designing bisulfite-specific primers to include the CPG sites, and for genetic markers, this was done by applying BNA and LNA clamps to inhibit wild-type amplification. We also developed and validated Fast NGS Detect, a software to expedite NGS data analysis. And lastly, we road tested this developed HCC-targeted NGS technology platform on 46 urine DNA samples. By successfully increasing the sensitivity and by successfully developing Fast NGS Detect, we proved our hypotheses.
The novelty of our project includes the approaches we applied in sample preparation to improve the NGS assay sensitivity, as well as our success in simultaneously detecting both genetic and epigenetic DNA markers in urine. Lastly, we developed a new software, Fast NGS Detect, that effectively detected modifications of the tested genes with a time saved greater than 100-fold. In the future, we plan to improve our assay by simplifying the steps by multiplexing. We can also achieve a higher throughput by potential automation. We plan to refine our software to include more features and make it fully customizable to both the public and to other types of cancers. And lastly, our assay and our software can be utilized in precision medicine and in liquid biopsy for cancer management. We would like to thank Dr. Hsu and Dr. Jane for their support, as well as the Siemens Foundation and George Washington University for this opportunity. Thank you.